Hello, this is a spectrophotometer. It is measuring photons. No? It's photons? Okay, all right. So we are going to demonstrate how to use the spectrophotometer. That is not going to be one of your responsibilities to actually know how to use it, but you should understand the data that we're getting from this. So basically what we're going to be testing is absorbance and transmittance of a sample, or that's absorbed from the sample of spinach that's been dissolved. So in order to use the spec, we need two cuvettes. Ta-da! The clear one is called a blank. The blank is used to calibrate our instrument. Right? It is going to get put into the spectrophotometer after we make sure we wipe off all the dirty fingerprints. Slides in. All right, and then zoom in, fancy Hollywood. We will hit the calibrate button, which is basically like zeroing out a scale or your calculator. And once it calibrates, woohoo, it shows us that there is a 0 absorbance, and we can also look at the percent transmittance, which should be 100%. Now we are going to put in our sample. Now the sample has been dissolved into alcohol. All right, that alcohol is going to give our spinach something to sit in. And when we put that in, you don't have to push anything, magic. All right, we get an absorbance of 0.869 at a 400 nanometer wavelength, and then the percent transmittance is about 13.5. Now, this trend that we're going to see is that as absorbance gets higher, percent transmittance is going to get lower, right? An inverse relationship. So we're not going to go through all of this because there's a lot of them. If you look in your lab book, you will see that you have a whole list of all of the wavelengths that are going to be tested. And then we made up some trendy data. We just made it up a little bit, that follows absorbance through the rainbow, basically. So if you look really closely, you can see that the percent transmittance is low when the absorbance is high, right? And that is very consistent through the top part of our number data. And then it gets really opposite of that, right? Percent T is high and absorbance is low. And then as you hit 660, it's the magic number, we get a spike in absorbance and a drop in percent transmittance. So if we look at what that correlates to on the rainbow, right? You have a high absorbance in the purples and the blues, and then at green, at about 500, we get a major drop in absorbance and an increase in percent transmittance. And then things stay pretty low for a while until you hit that magic 660 mark. Right? At 660, the percent T goes way down and the absorbance goes way high. So what does that mean for our plant? That means that the plant is using the purples and the blues for photosynthesis, not using as much of the green and the yellow and the orange, and then using a wee little bit of our red. Right? So the spectrophotometer, right, is going to help us to measure percent T and absorbance at certain wavelengths while using a plant dissolved in alcohol in order to be able to measure those values.